My project mainly focuses on the reproductive biology of cannas. So let me just give a bit of a background information about cannas. Cannas are all native in South and Central America. The genus is composed of 8 to 10 species and more than 1,000 hybrids. And these hybrids are thought to contribute to invasiveness. Today, the species is distributed across all the tropical and subtropical regions of the world, and most species are presumed to be self-compatible. And there is no data available for other species about their compatibility systems. So the genus has also many profound modification in relation to other clades. One of the profound modifications which I found interesting was the secondary pollen presentation, which is when the anther deposits its pollen to form a secondary pollen present. Why have I chose to do this study? It's because that in South Africa and around the world, cannas some species of cannas are declared invasive. In South Africa, for example, Canna indica L is declared invasive. And that most previous research about canna is limited to the nutritive and pharmacological aspects of it. So ultimately, there is little data available about the reproductive biology of cannas. And that now there is little data available about the reproductive biology of cannas, and because it is invading in Pietermaritzburg and Howick. So the study aims to investigate the reproductive trait that promotes invasiveness in Canna cultivars that grow in Pietermaritzburg and Howick. So the working hypothesis of this study was that the Canna cultivars that I'll be working with are self-compatible and can self-pollinate. This is basically what the study will test, and it will also examine whether there are suitable pollinators in this new region of invasion. So the methods that I've employed are based on just standard pollination experiments which were done in 3 cultivars, which is the red cultivar, the orange, red, and the yellow cultivar. So what I've set up breeding systems experiments. I've also set up compatibility systems test. I've also tested if these plants are affected by pollen limitation. I also test if these plants can reproduce vegetatively. And I've also checked if these plants have a suitable pollinator. That is where I would exclude the birds. What I have found so far is that in the breeding systems experiments, the fruit set is high, but there are no seeds that are set. In the compatibility systems experiments, I found that fruit set is high and seed set is also high, as well as goes to the pollen limitation test, but the fruits and seed sets are low. And when I exclude birds in my bird exclusion experiments, I found that fruits and seed set is moderate in comparison to the other tests. And I also found out that these plants can reproduce vegetatively. Thanks.